Okay, hello again. So we're down to the last sub-metabolic pathway involved in metabolism. That would be protein metabolism. And contrary to carbohydrates and lipids metabolism, protein metabolism is quite not very straightforward because of how proteins are denatured. Then they are formed into amino acids, and their amine groups are being transferred from one part to another, so forming either acetyl coenzyme A or carboxylic acid derivatives or even carbon dioxide or even the amino acid groups that we have discussed during our discussion on proteins. So, for protein metabolism, we are concerned of how proteins enter the body. They are being broken down to smaller counterparts before their amine groups are either transferred or removed. And we'll also talk about or discuss briefly why we have to eliminate amine in the form of ammonia and as it enters the urea cycle. So we're already familiar of how cycles work throughout metabolic processes in the body, especially or patterned like Krebs cycle. And then we'll go over two diseases involved in protein metabolism. We have phenylketonuria or MSUD or maple syrup urine disease. So when proteins enter the body through the mouth, so from there, digestive enzymes already welcome these protein-rich foods. And when they enter the stomach, so they are greeted by pepsin and hydrochloric acid. Then the linings of the small intestine and the large intestines have enzymes and hormones that help synthesize the amino acid from large molecules or the polypeptides. Okay, so these are the different enzymes found in the lining of the intestines. Here we have secretin and CCK or cholecystokinin. And the pancreas secretes the following. So trypsin, chymotrypsin, and elastase. Now sodium bicarbonate neutralizes the hydrochloric acid and food mixture. Okay, so how does the body break down proteins that are for consumption from the proteins that make up the organs? So simply put, so those enzymes released are inactive. They are only activated when they already enter the small intestine. So katong trypsin, chymotrypsin, they are stored as inactivated counterparts. So they are stored as trypsinogen and chymotrypsinogen. Then when they are entering the small intestine, so another enzyme called enterokinase. So entero in enter, and kinase, so phosphate or binding energy. It binds to trypsinogen and then it is converted into trypsin. Then this trypsin then binds to chymotrypsinogen to form the active chymotrypsin enzyme. Okay? And then that's where these two enzymes, trypsin and chymotrypsin, then break down the proteins into smaller peptides through hydrolysis. And that process of breaking down large proteins into their smaller peptides is called proteolysis. Okay, so the technical term or the word to describe gastric juice and food mixture is called chyme. And as mentioned earlier, sodium bicarbonate neutralizes the hydrochloric acid in the stomach or in the mixture. Okay, so in other words, our stomach is, well, acidic, di ba? But it is just enough, or it is basically the lining of the stomach is protected by the enzymes and hormones that will not be penetrated by HCl. Okay, so that's why also when there are, say for example, contaminated food, so our stomach does not rupture, does not get damaged right away. That's because of HCl. So there is this video about under the microscope in a slide, there are parasites and HCl. Then apparently, the hydrochloric acid can actually kill or inactivate or perhaps damage the parasite. However, it is not enough to kill or to damage the eggs inside the parasite. That's why it is still important that the temperature of the stomach is optimal and that we cook our food at the right temperatures. Because even though the parasites will die, their eggs might not. Their eggs might survive. Okay? And those parasites might continue to live on. So that's another downside despite the stomach being acidic in nature. So there are two reactions involved in how amine groups or amino groups in proteins are being converted into urea. Okay, because different amine groups are found in the different parts of the metabolic pathways. So, like I've mentioned earlier, if glycolysis and lipid metabolism are straightforward, amine on the protein metabolism on the other hand is marag iskina siya. So any part of the metabolic pathway where there are amino acids, it is automatically considered protein metabolism. So there are two ways. It is either transferring of amino groups or transamination from one nitrogen containing molecule to another molecule, or deamination. So that is totally removing the amine group to the amino acid used for energy or perhaps to become urea. Then both of these reactions have the same goal: to convert the amine group for energy and to convert the excess or particularly ammonia into urea so that it will be excreted through the kidneys or in urine. Okay? So one particular example would be the transferring of the amino or amine group or ammonia group in alanine to glutamate through the help of the enzyme alanine transaminase and the other molecule, your alpha ketoglutarate. So if you recall, I have mentioned that in the citric acid cycle, the alpha ketoglutarate is an entry point for protein metabolism because it can aid in transferring the amine group from alanine to glutamate or to form glutamate, and this glutamate will be oxidized, oxidative deamination, to separate, forming alpha ketoglutarate and urea. On the other hand, alpha ketoglutarate will transform into pyruvate, and this pyruvate is the same pyruvate that can be used to oxidize to form acetyl coenzyme A, and then the cycle goes on again. Okay, so in other words, that's how protein metabolism is found almost everywhere because of its intermediary forms of molecules. So oxidation is always involved in these reactions to form their counterparts. So it is to degrade these protein derivatives so that we'll be able to obtain pyruvate and urea. Okay, so for proteins, byproduct, although byproduct is urea, so still it is considered an important product. amino acids can either be categorized depending on which uh, correlative metabolic pathway they can be converted into other important molecules. So if amino acid can be converted into glucose through gluconeogenesis, so we consider them glucogenic. On the other hand, if this amino acid can be converted into ketone bodies, then later on, so ketone body will become fat, as mentioned in the lipid metabolism, then we call these amino acids ketogenic. So here, like I mentioned, murag kada eskina, protein metabolism is present. Okay, so based on this diagram, so for glucogenic, it's in red. The ones in green are ketogenic. And if you notice, so there are only two that are purely ketogenic. You have lysine and leucine. And then those in violet are both glucogenic and ketogenic. So we have a lot. And for the least ones are ketogenic, meaning to say only a few amino acids can be converted into ketone bodies, and the rest they are more of they are a mixture of both. Then there are more purely glucogenic amino acids than purely ketogenic. So why does the body have to eliminate ammonia in the form of urea? So the reason is simple. It is toxic. So if you recall how the body metabolizes alcohol, so alcohol is toxic, so it has to be detoxified, or the red blood cells that have been affected by alcohol has to be detoxified before the body is able to eliminate it. So that's the same idea here for converting ammonia into urea, still in the liver, before the body will eliminate urea in the form of urine. So urea here is already considered non-toxic. If it is from the liver, then to the kidneys, then in the urine. So that is the work of urea cycle. So it is still a set of reactions that produces urea from ammonia in the form of ammonium ions to prevent toxic levels of ammonia or ammonium in the body. So it is primarily in the liver, although the one that generates urine is the kidney, or are the kidneys. But the one that converts ammonia into urea is the liver. So ammonia contains waste nitrogen. So as I have mentioned in the earlier discussion on metabolism, our body has to eliminate nitrogenous wastes in the form of ammonia, then urea. So this is it. This is the cycle that is able to detoxify the liver of ammonium ions so that it will be eliminated in the kidneys, then in the urine. So from here we can see that ammonia is simply converted into its corresponding intermediates before it becomes urea. So these enzymes are more of synthesis and lysis, so breaking down. We don't see much oxidation here, but rather it is more of synthesis of or phosphate, synthesis of ammonia to form different phosphate counterparts before it becomes urea. So the last step, so if sa first step you need carbon dioxide, so dungan si carbon dioxide ug si ammonia, then here in the last step there is water to bind with arginine to form urea. Then this urea will travel through the bloodstream towards the kidneys, and the kidneys will then generate urine. Okay, so that's for protein metabolism. So we have proteolysis, breakdown of proteins, then transfer or removal of amine groups. We have transamination and deamination. Then we have the urea cycle, conversion of ammonia into urea. Then the urea will then be excreted through urine. Two diseases associated with protein metabolic disorder. We have phenylketonuria and MSUD. So PKU, MSUD. These are both involving deficiency in the enzyme present. So for phenylketonuria, the lacking enzyme is phenylalanine hydroxylase, meaning to say, children who are born with PKU don't have phenylalanine hydroxylase. Therefore, there could be buildup of phenylalanine in their bloodstream. So, one way to determine is in newborn screening. The newborn screening is really important so that the child will survive possible disorders at birth. So, it is genetic, meaning to say it is passed down from its ancestors having that gene. Then that child probably is not lucky enough in the gene pool. So there are different ways to treat, and one of which is to reduce protein intake, or perhaps avoid high protein. So that is to say their diets are restricted. The other hand, for maple syrup urine disease, so ganina, it's still enzymes, this one also. So, enzymes gihapon. So, for maple syrup urine disease, so the reason it's called maple syrup urine, it's because it is found that children born with this disease has their urine smelling like maple syrup. So sweet smelling. So apart from their urine, even their earwax or secretions, okay, so it smells like maple syrup, and they lack branch chain amino acid enzymes. So these enzymes are actually important, no, for growth and energy. So meaning to say, if they don't have these enzymes, mag-build up ang amino acid, so peptides, then health problems. So those are the two diseases. So both are found at newborn screening. So that's why newborn screening is essential for everyone. So as much as possible, so as early as possible, so every newborn must be screened to prevent further disease or even death. Okay. So that's exactly why metabolic disorders are ironic, because remember that metabolism exists to sustain life. Because of these disorders, so if there's no proper intervention, the children will die. So that concludes our discussion on metabolism, and here is a summary. So a diagram that summarizes everything from carbohydrate metabolism to the Krebs cycle, then ketones, lipids, amino acids, then all these processes go down to the electron transport chain, but this will is no longer part of our discussion since it's pretty much abstract. But the main point of electron transport chain is how electrons are being transported in the body. So how energy is being linked with these electrons. So it is also there where oxidative phosphorylation is being elaborated, and we all know that phosphates are the energy releasing molecules throughout the metabolic pathways. So here in this diagram, so we see that the darker green arrows show the catabolic processes, then the lighter green arrows anabolic. Okay, so this sums up everything. So see you in the exams and good luck.